Hi everyone. Good afternoon, guys. Welcome back to Unacademy NEET English channel. My name is Tina and I'm your biology educator. And yes, this is important. You must know that 21st June is approaching. Admit cards have been released. Please try to download them as soon as possible. And yes, guys, now it's high time that in this last 1 week you go on practicing uh, these tests at least twice a day, at least once at 2 to 5 p.m. and other at your convenient time. So make sure that you take this Unacademy All India Test Series. You need to know what to work at now. Today in this session, guys, I'm going to complete with class 12 NCERT reading. So we have already started that. Yes, please. So 4 chapters were done. In this session, guys, I'm going to start with molecular basis of inheritance, and let's quickly jump onto this. Quickly, let me know that you're all set. Yes, please. Shall we start? Give me a thumbs up. We're all set for the test. For this session. Your NCERTs are ready. All set? All righty. Yes, guys, good afternoon. So please share the session. Don't forget to like, subscribe, and share. See, when you like the session, it is a huge motivation and appreciation for us and gives us a lot of energy to work even harder. Anyways, so guys, this is molecular basis of inheritance, and we know what is the molecular basis, which is the chemical principle of inheritance. That chemical basis of inheritance is DNA, right? So starting with DNA is a long polymer of nucleotides, right? What is the length? Length depends upon the number of nucleotides or the pair. Now, in case of bacteriophage, you have to remember this. In bacteriophage, that is 5174, there are 5386 nucleotides. So if this question comes up in match the following, and that is already a PYQ, by the way. So if this question comes up, how do you have to remember this? So for 5 and 5, so that's how you remember. 5 has 5386 nucleotides. So having nucleotides means it has single stranded DNA. Then lambda bacteriophage. So lambda has 48502 base pairs. Those which have base pairs, they have double stranded DNA. E. coli has 4.6 into 10 raised to power 6 base pairs, and human DNA 3.3 to 10 raised to power 9 base pairs, and this is the haploid content. Now moving on to the structure of polynucleotide, we know that it is made up of nucleotides. DNA is nothing but a polynucleotide chain, and it has 3 components, which is nitrogenous base, pentose sugar, and a phosphate group. Ribose sugar in case of RNA, deoxyribose in case of DNA. 2 types of nitrogenous bases, purines and pyrimidines. We know this common. We know this. Then cytosine is common for both DNA. It's not just cytosine, it is A and G and C. All 3 are common for both DNA. Thymine is only present in DNA. Uracil is only present in RNA. Is that clear? I have just completed my special class, guys, and in that also I have revised the questions of genetics. My other special class will be tonight at 9 p.m. where we'll be continuing with the questions of genetics and others. Now, nitrogenous base is linked to OH of one carbon pentose sugar through N-glycosidic bond to form a nucleoside. And when these such as adenosine or deoxyadenosine and all these names, when phosphate is linked to OH of 5 carbon of nucleoside with a phosphodiester bond, it leads to a nucleotide formation, and you should know the names. Now 2 nucleotides are linked through 3-5 phosphodiester bond to form a dinucleotide, and many nucleotides will be joined in a similar way. Now there was this question just now I've discussed in the special class. Which of the following bond is responsible for anti-parallel nature? Let me know the answer. Which bond is responsible for the anti-parallel nature, guys? Which bond is responsible for anti-parallel nature of DNA? Anyone who can answer that? Ma'am, Yes, please. When there will be MCQ practicing session, Vishali have just taken at 11 p.m. on Unacademy Learners app. Special classes are free, bache. Nobody knows the answer. Hydrogen bond, no Akshay. Anti-parallel nature is because of phosphodiester bond. In both the strands, it has opposite polarity. Now, the phosphate moiety, which is referred to as this. Similarly, at the other end of the polymer, sugar has a free OH group, which is referred to as 3' end. Phosphate moiety 5' end, which is referred to as 5' end. Now, backbone is due to sugar and phosphate. Now, this is common for both DNA and RNA. Nitrogenous bases linked to sugar project from the backbone. Now, talking about RNA, RNA has additional OH group present at the 2 position, and it is the 2 position of sugar that will decide whether it is deoxyribose or ribose, right? Now, in RNA, uracil is found at the place of thymine. What is another name for thymine? 5-methyl uracil. This was a question I've just discussed. DNA is acidic due to phosphoric acid, and acidic nature was first identified by Friedrich Mischer. It was named as nuclein. Who named it nuclein? Mischer. Then due to technical limitations, the long polymer elucidation was kept for very long. Later on, 1953, James Watson and Francis Crick gave, based on X-ray diffraction data, X-ray was done by Maurice Wilkins and Rosalind Franklin, and then they gave the famous double helical model of DNA. What does this model explain? This represents the secondary structure of DNA. Secondary structure is 2-dimensional structure. Is that clear to all? Then there were propositions. This structure was also based on the propositions by Erwin Chargaff. And this Chargaff's rules are therefore applied for only double-stranded DNA. They don't apply to single-stranded DNA. Is that clear? Ratios between A and T, G and C are constant or equal. This is, now remember here, A plus T and G plus C. This ratio is constant for a particular species. So every species will have a different ratio, AT plus GC, while AG upon TC, AG plus TC, this is equal to 1. This is purine upon pyrimidine. Is that clear? Please be clear about these ratios, which ratio is constant and which ratio is equal to 1. The base pairing is a unique property. Now this base pairing formula, this base pairing rule, will apply to DNA formation, that is replication. It will also apply during transcription, DNA to RNA. It will also apply when mRNA binds to a complementary tRNA. Is that clear? But it does not apply to amino acid synthesis. Amino acid binding to the, it doesn't apply to the genetic code. Simple as that. Is that clear to everyone? Now, yes, please. What are the salient features of the double helical structure? The salient features, there are 2 polypeptide chains. The backbone is formed by sugar phosphate, bases project inside. The 2 chains are anti-parallel, one in 5-3 direction, other is in 3 to 5 direction. And similarly, there is different polarity of phosphodiester bonds also. Now, the bases in the 2 strands are connected through hydrogen bonds. Hydrogen forms are formed between the 2 nitrogenous bases. A and T form 2 hydrogen bonds, and G and C forms 3 hydrogen bonds, right? So purine always comes in front of pyrimidine. This maintains uniform distance between the 2 strands. So basically, uniform width is maintained here. We have just solved these questions. Now the 2 chains are coiled in right-handed helix. It is a right-handed helix. What is the length of the helix? 3.4 nanometer. They can also call it pitch or loop. Is that clear? So if they say 20 loops, so that means 20 pitches are being discussed, pitch or loop. And how many base pairs per loop? That is 10 base pairs. We are talking about bDNA. Is that clear? Now the distance between any 2 base pairs is 0.3. We can call it rise per base pair. So distance between two. Now the plane of one base pair stacks over the other in a double helix. In addition to this, confers stability, and this confers stability to the helical nature, the plane of the base pair. Is that clear to everyone? Now, the proposition, another given by only Francis Crick, gave central dogma. It is the linear flow of information, the genetic flow of information. They gave linear. This Francis Crick gave the linear flow of information. Either DNA will form DNA through replication, or it will form mRNA by transcription, and then mRNA will form protein through transcription and then translation. So linear flow was given. Then later on, it was Temin. Can you suggest a simple nature for the process? Okay, Temin was someone who suggested that mRNA can also form DNA. He observed it in some viruses, reverse direction of flow, right? What do you call this process as? This process is called reverse transcription. Are these points clear? Then packaging of DNA, the distance between, see here, the distance between, yes please, hello. Who's this? From where? All right, guys. Now coming back, the distance between two consecutive base pairs 0.34 nanometer. Just read. It is a rise per base pair. Now see the length. How do you calculate the length? So length of DNA is calculated by multiplying the total number of base pairs with the distance between them. This is the formula. So this gives you the total length. Is that clear? For example, in humans, we have 6.6 into 10 raised to power 9 base pairs, the diploid content. And what is the distance between the two consecutive base pairs? That is 0.34 nanometer. So multiply 10 raised to power minus 9 and you will get it in meters. So what is the value of total DNA? That is 2.2 meters. Length is far greater. What is the space present between within the nucleus? It is 10 raised to power minus 6 nanometer. So now can you calculate the length of the E. coli? Yes, if the length of the E. coli is 1.36 nanometers, so length is given, can you calculate the number of base pairs? You will simply divide it by 0.34 nanometer. Is that clear or not? Yes or no? In prokaryotes, guys, since the DNA is in prokaryotes, there is no defined nucleus, DNA is not scattered, but it is held by some proteins which are positive charged, and that is when they form a region known as nucleoid. So what is the difference between nucleoid and the genetic material here in prokaryotes? Nucleoid is a region to which the genetic material extends, right? And it is these positive proteins, they are organized in some large loops. This DNA is held by some large loops held by the proteins. Is that clear? These are hook-like structures. These proteins are hook-like structures. Now see the question here for 2026, set of positively charged proteins are called histones, and they are rich in which amino acids? Lysine and arginine. Histones are organized to form, how many histones are organized to form octamer? 8 histones. This was 25-26. Now see the questions that have come up. The DNA is negatively charged around the positively charged histone, and that forms a stable structure called nucleosome. And how many base pairs are present in a typical nucleosome? 200 base pairs. Now the repeating unit of this DNA or in the chromatin is the nucleosome. Are we clear? Now nucleosomes are seen as beads on the string. This term is used for nucleosomes. Is that clear when you observe it under microscope? Yes, please. Now see, the packaging of chromatin at higher level requires additional set of proteins that are collectively referred to as non-histone chromosomal proteins. Loosely packed DNA is known as euchromatin, asked in 25, and more densely packed This is known as heterochromatin. What is the difference? Euchromatin is transcriptionally active while heterochromatin is inactive. That means euchromatin will form mRNA and later proteins, and heterochromatin will not form mRNA, and therefore no proteins are synthesized here. Is that clear? Now, search for the genetic material. The first experiment was done by Frederick Griffith. What is the name of the organism? Streptococcus. In case Streptococcus is not given, Diplococcus can also be the answer here. Now, this is responsible for causing pneumonia. Yes. Now, or pneumococcus bacteria also you can call it. There are two strains. Now remember, there is this S strain which is smooth and shiny, and there is R strain which is rough. Smooth is smooth because of glycocalyx layer, and now written here S because has mucus polysaccharide coat. Which coat are they talking about? Glycocalyx. They are talking about glycocalyx. R doesn't have. Now this glycocalyx or R strain is virulent, so bacteria, S strain bacteria will cause death, or it will cause the disease, right? While if mice is infected with this, it will die because of S strain. R strain does not develop pneumonia, so the mice will live. Now these 4 points are very, very important, these experiments. R strain is virulent, it is going to kill the mice. S strain. R strain is non-virulent, hence the mice will live, right? Now in the third experiment, S strain but heat killed was injected. Now when the S strain is killed itself, the mice will live. So this was the experiment. This was the experiment which confirmed transformation. So the 4 experiments will be given, all these 4. Which of the following experiment showed transformation? This was the experiment which showed transformation. See here, heat killed S strain when kept with R strain, live R strain. When injected into the mice died. And when this dead mice was studied further, so it was live S strain bacteria which was extracted out of it. From this dead mice, we extracted live S strain. Although we added heat killed S strain and R strain was live, then how did we manage to extract live S strain? So this confirms transformation. So this S strain has transformed the R strain. Please tell me these points are clear. Yes guys, are you able to follow the points? Yes guys, are you able to follow the points? Follow the points. Not audible. Vasant, what happened? Please check your mic. Yes guys, next is, so he recovered live S bacteria from the dead mice, and that's how he showed that transformed R strain have been transformed by the heat killed S strain. So this was known as the transforming principle, but he could not explain the biochemical nature. Biochemical nature was explained by these 3 scientists, Avery, MacLeod and McCarthy. Is that clear? He explained that genetic material was thought, which earlier was thought to be protein, but it is not so. The biochemical nature was worked through this experiment, and they also used the same setup as that of Griffith. Is that clear? And concluded that DNA is the hereditary material. Now, the unequivocal proof, that was a question in 2025. That means the ultimate proof was given by Hershey and Chase that DNA is the genetic material. What was the organism used? They used this. They worked with viruses that infect bacteria. That means they worked with bacteriophages. Is that clear? So the first step in their experiment was, so there were 3 steps. The 3 steps followed were here: infection, blending, and then centrifugation. Just remember that in order. So the 3 steps were infection, blending, and then centrifugation. See here. So first of all, during infection, the virus, they grew some virus on a medium containing radioactive phosphorus. When the phosphorus was radioactive, see, it led to radioactive DNA, right? And similarly, this radioactive sulfur made the protein radioactive. The direct statements can come up for your exam. Is that clear? Now, radioactive phages were allowed to attach and then infection proceeded. This is the first step. Viral coats were removed and this agitated in a blender. So, blending is the second step. And finally, followed by centrifugation. Now, see here. So you should be able to identify all these things. Now, properties of genetic material. So we have to remember 4 properties of genetic material. It should be replicable. It should be stable. It should have some chances of mutation. It should be mutable, which is important for evolution. And then it should be able to express itself in the form of Mendelian characters. So RSME, Rasme, you have to remember what are the 4 characteristics. Is that clear? Now, what is the reason behind DNA being more stable? See here, DNA chemically is less reactive and more stable as compared to RNA. It is because of different reasons. Number 1, the sugar, thymine is more stable. Second, the deoxyribose is more stable, and third, DNA is generally double-stranded. So double-stranded nature also provides it more stability. Are we clear? Now see, so these are the reasons. So both DNA and RNA are able to mutate. In fact, RNA is little more unstable and mutate at the faster rate. So there is this line. See here, DNA being more stable is preferred for the storage of genetic information and transmission. RNA is better. Is that clear? Now, RNA was the first genetic material. This whole paragraph was in 2025 statement-based question. RNA was the first genetic material. Suggests that essential life processes like metabolism, translation, and splicing evolved around RNA. RNA used to act as genetic material as well as catalyst. Now, being a catalyst also provides it less stability and more reactive nature. So, it is important biochemical reactions in the living system. RNA being a catalyst was reactive and hence unstable. This line. DNA has evolved from RNA and with chemical modifications which have made it more stable. DNA being double-stranded and having complementary strand resists the changes by evolving a process of repair. Are these points clear? Then this line, that replication, this line was given by to quote their original, Watson and Crick. They explained that it has not escaped our notice that specific pairing we have postulated immediately suggests a possible coping mechanism for the genetic material. So what are we talking about? We are talking about base pairing, and I already suggested you that base pairing is going to occur in, we are going to see that base pairing during 1, replication, 2, transcription, and 3rd, when tRNA binds to mRNA. The genetic code will not follow the base pairing rule. Is that clear? Now, what is this replication? What is the nature of replication? It is semi-conservative in nature. Is that clear? Now, the semi-conservative nature of DNA, the experimental proof in prokaryotes, first of all, was seen in E. coli, and this was suggested by Maselson and Stahel. But not in case of eukaryotes. In eukaryotes, the semi-conservative nature was explained by Taylor in Vishya Faba. Is that clear? What is the name of the experiment? What is the name of the experiment here? The name of the experiment here is cesium chloride density gradient centrifugation method. See here. So this was the experiment. So he grew E. coli in a medium containing N15 ammonium chloride. Is that clear? So it will incorporate and make it heavy DNA, which can be easily distinguished from the normal DNA by centrifugation in cesium chloride. So cesium chloride density gradient centrifugation method. N15 is not radioactive, rather it is a heavy isotope. Is that clear? So they are not going to check the radioactivity here. They are going to check the density here. Is that clear to everyone? They are going to check the density here. So see, in the beginning, when it was grown in N15 ammonium chloride, in that case, DNA was heavy. Both the strands had N15, N15, and see the bar of DNA that we have got. We have got it at the bottom of the test tube. And when there was this hybrid DNA, the density was slightly lesser. So it was in the upper layer. And then we got few light and few hybrid. So we got the two strands. Now, no matter what, we get some questions, numericals from this topic. No matter how many replication cycles have occurred, the number of hybrid strands will always remain 2. Is that clear? That will always remain 2. What is the replication time? That is 20 minutes. Is that clear to all? If you want to practice some more numericals with me, you will have to come for the special classes, guys. Now, E. coli was allowed to grow for 80 minutes. Then what would be the proportion of light and density, hybrid density DNA molecule? See, without looking at the minutes, hybrid DNA molecules will always be 2. Now, the question is, how many light strands were formed? So, for this, see, we know that replication time is 20 minutes. So, in 80 minutes, how many replication cycles would have taken place? How many? So, 3 replications would have taken place. So, how to calculate the total number of strands, DNA strands formed? That is equal to 2 raised to power n, and n is the number of replication cycles. So, 2 raised to power 3, how many total strands have been formed? 8. If out of 8, 2 are hybrid, how many light have been formed? It will be 6. Is that clear? So, this is how you calculate. Now, here it's clearly written in Vishya Faba. See, Taylor demonstrated that DNA in chromosomes also replicate semi-conservatively. The organism used was Vishya Faba or the Faba beans. So, Taylor established the semi-conservative nature of DNA replication in eukaryotes. So, read the question properly, whether they are asking for eukaryotes or prokaryotes. Is that clear? Now, coming on to the machinery of enzymes. Machinery and the enzymes, see. Yes, for replication, first of all, what do you need? The main enzyme is DNA polymerase, and it is DNA dependent. Is that clear? So, what is it employed for? For polymerization of deoxyribonucleotides. Now, the process of replication in case of E. coli, we have just read is 20 minutes, but here it is written within 18 minutes. So, this is to be precise, but for calculative purposes, we consider it 20 only. So, the average rate of polymerization here is around 2000 base pairs per second, and this is done with very high degree of accuracy. Is that clear? Now, energetically replication is very expensive process. Deoxyribonucleotide triphosphates serve a dual purpose. Number one, it acts as a substrate. Also, when the bond breaks, it provides energy also. So, deoxyribonucleotides will provide 2 functions. Is that clear? Now, next is, in addition to this polymerases, there are other enzymes required. The 2 strands cannot be separated the entire length. Why the 2 strands cannot be separated in one go? Because it demands a lot of energy. Therefore, it opens within a small opening known as replication fork. Now remember, this polymerase enzyme will always read in 3 to 5 direction, and it will polymerize in 5 to 3 direction. See, polymerization only in one direction. It is only in one direction, right? And it reads in, that means it attaches itself in 3 to 5 direction. This is the rule. Whether it is DNA polymerase or whether it is RNA polymerase, it will always read in 3 to 5 and always polymerize in 5 to 3. Is that clear? So, here the word is only or always. Now, see, there are both the strands will act as template strands. So, the polarity 3 to 5 will be a continuous strand, and the other strand with polarity 5 to 3 is a discontinuous strand. And in discontinuous strand, there are fragments formed which are combined by the enzyme DNA ligase. Is that clear? Now, replication does not initiate randomly, rather it occurs at a special place known as origin of replication. Is that clear? Now, because of the requirement of origin, the vectors provide the origin of replication. Now see here, when does replication occur? Replication takes place during S phase of cell cycle. What will failure in cell cycle division lead to? Failure in cell division will lead to polyploidy or the chromosomal anomaly. We have read this line in the previous chapter also. That what leads to polyploidy? Failure in cell division. That means no cytokinesis. Now see, we have continuous synthesis, and towards this strand we have discontinuous synthesis. So this is known as the lagging strand, and this one is known as the leading strand. What is the polarity of the leading strand? We have just read, we have just read it here. The leading strand is continuous. Continuous is 3 to 5. So this is 3 to 5, and this is 5' to 3' direction. Now overall replication, what is the nature of replication? Now see, the overall replication is semi-conservative. Why? One of the strand is parental, and it is also semi-discontinuous. It is also semi-discontinuous. Why? It is continuous on one strand and it is discontinuous towards other strand. So we don't call it a discontinuous, it is semi-discontinuous. Remember these 2 points as well. Is that clear? Now, transcription. Transcription is copying of genetic information from DNA to RNA. Again, it follows the principle of complementarity. Rule of complementarity is followed here, just that uracil instead of thymine will be used. Is that clear? Now, the transcription unit. This question was already asked in 2026 exam. The transcription unit is made up of promoter, structural gene and terminator. Yes. Now, the two strands have opposite polarity and the enzyme used is DNA dependent RNA polymerase. Again, polymerization only in one direction and the polarity, and it will read always in 3 to 5 direction. Now the polarity 3 to 5 acts as a template strand, and the template strand will be known as template strand or non-coding strand. And the other strand with polarity 5 to 3, this is a coding strand. Coding is the non-template, while the template is also known as non-coding. So don't get confused, we are using a opposite terminology for this. Is that clear? Now see here, how do you determine which of them will act as a coding strand or the template non-coding strand? So always look for the position of promoter. Now the one which contains, so the side which contains promoter, the strand which has 3' end, this will act as a template strand. Is that clear? But now what is the use of coding strand? That means the opposite, the non-template strand. This non-coding strand, this coding strand or the non-template strand will act as a point of reference. This is used as a point of reference. Now see, 2026 had so many questions from this paragraph, and it is actually very important. Promoter is said to be located towards, where is promoter located? Promoter is located towards 5' end upstream of the structural gene. Is that clear? So reference is made with respect to the polarity of coding strand. This is what I told you. It is a DNA sequence. What is promoter? It is a DNA sequence that provides binding for RNA polymerase. So presence of promoter. So I told you who's going to define which one will be template and which one will be coding. So presence of promoter defines the template and the coding strand. These were all lines here in 2026, and questions may be repeated, guys. Terminator. If we talk about this terminator, located towards 3' end, and 3' end is known as downstream of the coding strand and defines the end of the process of transcription. Are these points clear to all? Right? Now, what is a cistron? Cistron is nothing but a gene only, but that gene or that part of DNA which codes for RNA, right? See, mRNA, the segment of DNA coding for a polypeptide, and the structural gene is a transcription unit called to be monocistronic. Monocistronic is found in eukaryotes, while polycistronic is a nature of prokaryotes. So we have PP. Is that clear? Now in eukaryotes, monocistronic structural genes is interrupted by some non-coding parts which are known as introns. So monocistronic will have the expressed sequences called exons, and it will also have introns. Introns are the intervening sequences which don't express themselves. So these are the two components of monocistronic structural genes, or called, and this is why it is known as split gene arrangement. So split gene arrangement is a feature of, this is a feature of eukaryotes only. Prokaryotes don't have split gene arrangement. So we are talking, so eukaryotes have monocistronic, both exons and introns, and this arrangement is known as split gene arrangement. Please tell me these points are clear. Now apart from this, guys, there are some regulatory sequence, loosely defined regulatory genes that do not code for any RNA or protein, but they are essential for transcription. Now see, these are the three steps. One is initiation, second is elongation, third is termination. So question in 2025, which regulatory sequence is required for termination process? It is rho. Can they ask which regulatory sequence is required for initiation? It is sigma factor. So in initiation, RNA polymerase will bind to sigma factor. For elongation, it is only the RNA polymerase. Okay, it is only the RNA polymerase. And for termination, RNA polymerase plus rho factor will stop the process. Is that clear? Now, is that clear to everyone? So this is what I have already explained. So these termination factor or the rho factor and initiation factor, that is the sigma factor, it associates temporarily to initiate or terminate the process. Is that clear? Now, so association with these factors alter the specificity. They alter the specificity of RNA polymerase, whether it is going to initiate or terminate. So the same enzyme will terminate as well as initiate depending upon these regulatory sequences that they are binding to. Are these points clear to everyone? Yes, please. Are these points clear to everyone? Right? Now, see, in bacteria, this transcription and translation is going to occur in the same compartment, that is in the cytosol. That will occur in the cytosol because there is no compartmentalization, right? Now, in eukaryotes, guys, in eukaryotes, there are two additional complexes that will be formed. First of all, in eukaryotes, it occurs within the nucleus only, right? Because now there is clear-cut compartmentalization, there is division of labor. And another difference, in prokaryotes, be it tRNA, mRNA or rRNA, all the different type of RNAs will be formed by the same RNA polymerase. But here in eukaryotes, we have three different type of polymerases. RNA polymerase 1 transcribes rRNA, 28S, 18S, 5.8S, while the 3 is responsible for tRNA, 5S RNA and snRNAs, while the 2-1, it codes for mRNA and hnRNA. So these points are important and can come for match the following. Then another question. Second complexity is that primary transcript is hnRNA, contains both exons and introns. Is that clear? So we need to cut these introns. We need to remove these introns, and this process is known as splicing. Another, capping has to take place. So this was asked in 2025. So we have to cut the non-coding parts, that is the introns. Second, we have to protect the two ends, 5' end and 3' end of mRNA. So on 5' end, guys, on 5' end we add methyl guanosine triphosphate. This process is called capping. And then there is third process called tailing, which is done on TT. T for tailing, T for 3. On the 3' end we do tailing, guys. Is that clear? Where adenylate residues are added. Are these points clear? Now all these three steps, splicing, capping and tailing, this constitutes the post-transcriptional changes. These post-transcriptional changes are not required in prokaryotes. Is that clear? Please tell me that these points are clear to everyone. Now there is this line that in split gene arrangement, the presence of introns is reminiscent of antiquity. Antiquity means antique nature. It shows the evolution that these genes have been carried forward, antiquity, and the process of splicing represents the dominance of the RNA world. Is that clear? Now coming on to genetic code. Now genetic code tells you about the sequence of amino acids. Okay, right? So it was George Gamow, right, a physicist, who suggested that a code should be made up of 3 nucleotides. So basically, he deciphered the triplet nature of genetic code. This was by George Gamow, and this question was asked in 2025. Is that clear? Then this is your genetic code, which you don't really have to learn directly. Now, Marshall Nirenberg's cell-free system for protein synthesis helped code to be deciphered. Severo-Ochoa enzyme. Severo is in the name of the scientist. Severo-Ochoa. Polynucleotide polyphosphorylase. This enzyme was helpful in polymerizing RNA, which defined the sequences in the template independent manner. Is that clear? Now, what are the salient features of the genetic code? Triplet. This was suggested by George Gamow. How many codons are there? Total 64, out of which 61 code for amino acids, 3 codons do not code, and hence they are known as stop codons. Then the degenerate nature. What is degeneracy? That one amino acid can be coded by more than one codon. Is it clear? Then it is always read in continuous manner. There are no punctuations. It is comma-less. You can also call it comma-less. Nearly universal. Why nearly? Because there are some exceptions found in prokaryotes and in mitochondria and in protozoans. Then AUG has dual nature. It codes for methionine as well as it is a start or a initiator codon. And which are the 3 stop codons? You have to learn these names. Is that clear? Now, coming on to mutations. This line was in 2026. Point mutation is a change of single base pair in the gene for beta chain. This is an example. But point mutation is a change in single base pair in the gene. This is the definition. That results in the change in amino acid residue from glutamate to valine and results in a disease called sickle cell anemia. So we know what happens in sickle cell anemia. GAG is replaced by GUG. So instead of A, it is uracil. So this changes the triplet nature. It changes the amino acid it is coding for. This causes sickle cell anemia and it is an example of point mutation. What is point mutation? Where there is change in only single base pair. Is that clear to all? Now, what is frame shift? See, frame shift mutations occur because of insertion or deletion of 3 or its multiple. Hence, insertion or deletion of 1 or 2 base pair changes the reading frame, leading to frame shift mutation. But remember, if you add 3 or the multiples of 3 or you delete 3 or multiples of 3, this will not alter the, the reading frame remains unaltered. So if you add 1 or 2 bases or you remove them, it will alter the frame. But addition of 3 or multiples of 3 will not alter the frame. Is that clear? Now, talking about tRNA, tRNA is the smallest of all the RNAs. It is known as soluble RNA as well as the, soluble RNA also the adapter molecule. Clear to everyone? Now, tRNA has an anticodon loop. So this is the anticodon loop, right? Because on anticodon loop, we have this anticodon. So on mRNA, guys, on mRNA, we have codon. See, mRNA has codon, while tRNA has anticodon. Is that clear? And they also follow the rule of complementarity. They'll bind to it. So A complementary to U, G, C, U, A. So A, G, U complementary to U, C, A. So they are binding according to the principle of complementarity. Is that clear? Now, there is tRNAs are specific for each amino acid. So for initiation, there is another specific tRNA that is referred to as initiator tRNA. Like we have initiator codon, start codon, AUG. So similarly, for AUG, we will have UAC. So tRNA with this anticodon will bind to this. So this is the start codon. This is present on. This is the initiator tRNA. Are you getting this? Are you getting this? Now see the structure. And also, See, we have 64 codons, but we have only 61 anticodons because there are no tRNAs for the stop codons. Now, talking about the structure, the secondary structure, that is the 2D structure, is clover leaf, and the actual structure is always three-dimensional or tertiary structure, which is inverted L. Is that clear? So this is about tRNA. Now talking about translation, guys. So translation is a formation of proteins, right? So formation of proteins. So first of all, it is due to the polymerization of amino acids. Now the sequence and the order is defined by the sequence on the bases in sequence of bases in mRNA. Now amino acids are connected through peptide bond and which requires energy. Therefore, in the first place itself, amino acids are activated. Activation of amino acid means binding of amino acid with ATP. Is that clear? Now, this is known as, and then these activated amino acids are put on the tRNA. This is called charging of tRNA or amino acylation of tRNA. Is that clear? Now, when the two charged tRNAs are brought, there is formation of a peptide bond because this is favored by energy. Is that clear? Now the presence of catalyst is also going to enhance this rate of peptide bond formation. Let me know the answer. Which catalyst will enhance the peptide bond formation? Yes, guys. And it is not made up of proteins. Just a hint. Now, the ribosome consists of structural RNA. What is ribosome made up of? It consists of rRNA and some 80 different proteins. Is that clear? Now, when they have to get involved in translation, they have to bind to each other because otherwise they are moving, they are present separately. Is that clear? Now, the ribosome also acts as a catalyst. I told you, 23S rRNA in bacteria is the enzyme ribozyme which helps in the formation of peptide bond. Is that clear? Now, what does translation unit consist of? It is a sequence of RNA that is flanked by start codon, stop codon, and codes for the polypeptide. So, there are three units. Again, start codon, stop codon, and in between the codes for polypeptide. Is that clear? Then some additional sequences which are not coded, this is known as UTR. They are present on both the ends, required for increasing the efficiency of translation process. Are we clear? Please let me know. Are we clear here? Now, amino acids are added one by one, translated into polypeptide sequences dictated by DNA and represented by RNA. At the end, a release factor binds to stop codon, terminating the translation, and this completes the polypeptide. Is that clear? Now, what is regulation of gene expression? In eukaryotes, guys, regulation can occur at various levels. So, see, there are so many levels at which regulation of DNA can take place, of gene can take place. You can first not allow the mRNA formation, hnRNA. Even if it has formed, don't allow the formation of mRNA. So, regulation of splicing and post-transcriptional changes. If it has been formed, then don't allow mRNA to come into the cytoplasm. And even if it has come into the cytoplasm, you can still prevent translation from occurring. So in eukaryotes, we have several levels at which the regulation can be exerted, but not in case of prokaryotes. Why? Because prokaryotes, we know, first of all, direct mature mRNA is formed, and then transcription and translation is coupled together, right? So, in prokaryotes, control of rate of transcription initiation is the predominant site for control. If you don't want the protein synthesis to occur, you don't let the transcription to occur. Simple as that. So for that, guys, we have special system which is called operon. We have a proper model which is known as operon. So activity of RNA polymerase at given promoter in turn regulated by some accessory proteins. Like we have regulatory proteins, right? And then we have operators and we have promoters and so on. So this operator is adjacent, operator is always present adjacent to promoter element in most of the operons, and in case the sequence of the operator bind to the repressor protein. Is that clear? Now we are going to discuss lag operon here. Lag operon, it was studied by Jacob and Monod. It was studied by Jacob and Monod, a geneticist. Now, LAC word refers to lactose. It is a polycistronic structural gene with common promoter and regulatory, and it is found in bacteria, let's say E. coli. Is that clear? Let's say E. coli. Now, LAC operon consists of one regulatory gene, and this regulatory gene is denoted by I. This was a question in match the following. See that. I does not refer to inducer. I refers to repressor, in fact. I refers to regulator. Is that clear? And the word has been derived from inhibitor. Why do we refer regulator as I? Because the protein which will be produced by regulator, which is repressor, it is going to inhibit the formation of proteins. So word comes from inhibitor. There are three structural genes, ZYA. I gene, it codes for repressor. Z for beta. So we'll study all these from here only. See, this was a question. What does this Z code for? This was asked in this year itself. Z codes for beta galactosidase. Can they ask for Y and A also? Y codes for permease and A codes for trans-acetylase. Is that clear? So, and what does this regulator gene code for? Regulator gene codes for repressor protein, and this is an inhibiting protein. Where does it bind? It binds to the operator and prevents the RNA polymerase from transcribing the operon. Are we clear? Now see, lactose is a substrate for the enzyme beta galactosidase, and it regulates the switching on and off of the operon, hence it is a inducer. Is that clear? So if lactose is present, we want the enzymes to be produced, otherwise we don't want. Through permease, the lactose is transported into the cells through the action of permease. So these are the functions of each and every enzyme. Is that clear? Remember, at very low level, expression of lac operon doesn't work. It cannot enter. Lactose then induces operon in following manner, and we have studied that. By the way, instead of lactose, it can be allolactose also, which can do the same function. So they are isomers. So instead of lactose, it can be allolactose also. Is that clear to all? Now, remember, glucose and galactose cannot act as inducers, and regulation of lactose, lac operon by repressor, is referred to as negative regulation. It is also under the control of positive regulation, but it is not explained here. Now, talking about the Human Genome Project, see, it was launched in the year 1990, right? And it was a mega project of around 13 years. 13 years it took. Now, yes, please. So we can skip this part. Just remember this. It was closely associated with the rapid development of new area in biology called bioinformatics. What are the goals? The goal is to identify all these 20,000 to 25,000. But later on, we got to know there are around 30,000 genes in human. So determine all the sequences, 3 billion chemical base pairs, store this information in database, improve the tools for data analysis, transfer this technology to other sectors, and address the different issues. See, told you, 13-year long project, mainly coordinated by US Department of Energy and NIH, that is National Institute of Health. The major partner was Wellcome Trust of UK. Other additional partners, Japan, France, Germany, China, and others. So you should know the name of these countries. It is Japan, France, Germany, and China. Project completed after 13 years, that was in 2003. Is that clear? Now, so it helped us to study lots of human disorders as well. Right, besides providing clue for human, it also learning about some non-human organisms was also possible. Because of this, many non-human models such as this have also been sequenced. Anyways, now see the methodology. There are two methodologies here. Expressed sequence tagged, where we identify all the genes that are expressed, that is exons only. And then we have the sequence annotation. In sequence annotation, we study both coding and non-coding sequences, both exons and introns. Is that clear? Now, the commonly used hosts were bacteria and yeast, and the vectors were BAC and YAG. Is that clear? Now here, there is another scientist. The fragments were sequenced using automated DNA sequences, worked on the principle method developed by Frederick Sanger, credited for determination of amino acid sequences in proteins also. So this sequencing was done based on his principle. Is that clear to everyone? Now, the chromosome which was completed or sequenced in the end was chromosome number 1, the last to be sequenced. Clear? Let's talk about the salient features here. Human genome contains this much base pairs, right? Average gene size is 3000 bases, and the largest one is dystrophin with 2.4 billion. Total number of genes 30,000, which was previously estimated to be 80,000 to 1,40,000. Now, almost all the 99% nucleotide bases are exactly the same in all the humans. Function unknown for over 50%, less than 2% codes for the proteins. Repeated sequences make up the bulk of the DNA, and repetitive sequences are repeated many times and they have no direct coding function, just help in this. Now in 2025, this was asked that which chromosome has maximum genes. So you have to remember the number also, 2986. Can they ask about why? It is the smallest one and has the fewest genes, 231. Is it clear? So DNA is identified based on single base DNA differences, that is SNPs, single nucleotide polymorphism, that is SNPs. Is that clear? So that was application, and the major application is DNA fingerprinting. So fingerprinting is based on this polymorphism, guys. It involves identifying differences in some specific regions called repetitive DNA. Is that clear? It is a small stretch of DNA repeated many times and separated from the bulk as different peaks during the density gradient centrifugation. Now the bulk DNA forms the major peak and smaller peaks are referred as satellite DNA. Are we clear? So now we can divide it into microsatellites and mini satellites. So these sequences show high degree of polymorphism, which is the basis of DNA fingerprinting. So this is the principle, guys. The principle of DNA fingerprinting is polymorphism. Yes, guys. Are we clear? Are you reading with me? So it is a great identification tool in forensic application or in finding the paternity cases. Please tell me these points are clear to everyone. And also, see here, allelic sequence variation has traditionally been described as DNA polymorphism. Only if it fulfills two criteria, it will be known as polymorphism. See, DNA polymorphism. Number one, if more than one variant at a locus occurs in human population. This is criteria number one. And second, the frequency should be greater than 0.01. And this is inheritable mutation and referred to as DNA polymorphism. Is that clear to all? Is that clear to all? Yes, please. The technique of DNA fingerprinting was initially developed by Alec Jeffrey, and this question, the process has already been asked. You should be able to revise that and remember that. See, the previous paper was like a see-through into what's going in NTA's head. So if you go through the history, the questions have been repeated, even in Re-NEET exams, whenever they have happened. So make sure that you revise your 3rd May NEET 2026 exam or paper as well. Is that clear to all? Yes, guys. So with this, we complete this first chapter of molecular basis of inheritance. Please let me know that this chapter is clear. Yes, all the serious students here. All the serious students here, please let me know the chapter is done. Yes, please. All clear? Amrita, Swant. Venkatesh, there are some students who are facing this problem regarding the admit card. Please, first of all, try to do that in the morning or late night. prefer early morning. There are some students who are saying that they were able to download the admit cards in the early morning. Try that tomorrow. Okay, and make sure that you are filling in the details of the account first and then trying to download it. Without putting in the account details for refund, you won't be able to download the admit card. Is that clear? Yes. Now moving on to this chapter, evolution. See, what is evolutionary biology? Evolutionary biology studies the history of life forms on earth. Is that clear? Now, origin of life. So what are the stellar distances measured in the form of light years, not in meters or kilometers or miles? Then universe. How old is the universe? So this is the new data in case you are using the old book. So this is when the origin of universe happened. Origin of universe. 13.8 billion. Remember the value, it is billion. It is explained by Big Bang Theory, which explains the origin, and it was a single huge explosion. The universe expanded and the temperature came down. Hydrogen, helium formed after some time, and then in solar system, the Milky Way, this is the galaxy to which we belong. Earth formed around 4.5 billion years back with no atmosphere. So the primitive Earth had no atmosphere. Is that clear? Now, later on, see, ozone was formed and so on. Life appeared 500 million years after the formation of Earth, that is 4 billion years back. So you have to remember the sequence of events, also about other theories which explain the origin of life. So according to theory of panspermia, so spores came from different planets and gave rise to life. Then life came out of, this was first concept, then life came from decaying and rotting matter like straw. So this explains life came from non-living matter, abiogenesis, or theory of spontaneous generation. And then Louis Pasteur said life comes from pre-existing life, theory of biogenesis, used this flask experiment, round bottom flask experiment. Is it clear? So later on, spontaneous theory was also dismissed, however, could not answer how the first life form appeared. Why was it dismissed? Because could not explain how life came for the first time. Now, the theory that we actually trust is by Operin of Russia and Haldane of England, said that life could have come from pre-existing non-living organic molecules, and later on, the life now, so for present, life is coming from the pre-existing life only. But according to them, the first life came from the non-living matter. Is that clear? So abiogenesis first, followed by biogenesis. We have discussed it already. Conditions on Earth in the beginning, that was high temperature, volcanic storms, reducing atmosphere. So remember, what type of atmosphere was present in the early times or the primitive times? That was reducing atmosphere, right? In 1953, Miller, an American scientist, created similar conditions and explained and proved that life had this type of origin. So he created electric discharge in closed flask, and in 2020, guys, it was asked, see, which molecules were taken? It was methane, hydrogen, ammonia, and water was taken at 800 degrees Celsius. And what did he observe? He observed that amino acids were formed. He observed the formation of amino acids from just these 4 molecules. Is it clear? Now, we had no idea first replicating metabolic came from, we had no idea, but the first non-cellular form of life came 3 billion years back, could have originated. So this was the setup. See, these were the 4 molecules taken by Miller, and he generated this spark, and in the spark chamber, the reaction went on continuously for 17 to 18 days. Now, the first cellular form originated about 2000 million years ago. This version is called biogenesis. Is it clear? Once formed, how the first cellular forms of life could have evolved into the complex biodiversity of today. Now, evolution of life forms. The earth is about 4,000 years old according to only one theory which says life was created by God. So according to theory of special creation, earth is only 4,000 years old. But this is not the case. This is just a theory. Then, based on observations made by Charles Darwin, the name of his ship was HMS Beagle. He went to many places and see, he gave certain ideas. He gave us Darwinism, the theory of natural selection. See, another word for, he explained what is fitness of individual or population. Fitness according to Darwin refers to reproductive fitness. So those which are better fit in environment leave more progeny than others will survive more, and this is known as natural selection. Is that clear? This is known as natural selection. What are the evidences of evolution? First of all, we have fossils, remains of hard parts of life forms in rocks. Which rocks are we talking about? We are talking about sedimentary rocks, right? Now, different aged rock sediments contain fossils of different life forms. So very basic. So the upper strata will have more younger fossils, and it will have younger fossils, while deeper you go, it will have older fossils. Is that clear? Now, they represent the extinct organism. Can you name one extinct organism? Dinosaurs. Study of fossils is made in sedimentary layers. Yes. And all this is a paleontological evidence. How do you determine the age of fossils? We have different dating, radioactive dating methods like carbon dating, potassium argon method, uranium thorium method, and so many. Then embryologic. So this is the first evidence. Second is embryological evidence. This was proposed by Ernest Haeckel based on his observation that during embryonic stage, common to all vertebrates, they are absent in adult. Certain features are present only during embryonic stage. And he observed that embryos of all vertebrates, including human, develop a row of vestigial gill slits just behind the head, but it is functional only in fishes and not found in any other adult vertebrate. But it was disapproved by Ernest von Baer. Is it clear? So that was embryological evidence by Ernest Haeckel. Third, we have anatomical and morphological evidences. In anatomical and morphological evidences, guys, first of all, see, these examples are very, very important for homologous organs. So what are homologous organs here? Homologous organs are the ones which indicate common ancestry, right? So basically, they share similarities in the pattern of their structures. Yeah, see here, what are homologous organs? So they have common origin, therefore similar structure, but because these organisms have moved to different habitats, so they perform different functions. So if the two structures are given to you and you have been asked whether they show an homology or analogy, then the only quick way to answer such question is see their function. So different function indicates homology, while common function is going to indicate analogy. Is that clear? For example, see, bones of forelimbs in all the mammals, whales, bat, cheetah, and humans, they will share similarity in the pattern of their bones, but they perform different functions. They have similar anatomical structures, same bones are present. Is it clear? Second line, homology indicates common ancestry. This has already been asked. Then wings of birds and flipper of whale, they indicate homology. This was asked in 2016. Modified forelimbs. Clear? Then coming on to another example, the thorn and tendrils of bougainvillea and cucurbita. While if we talk about analogy, the first example is wings of butterfly and wings of birds. All these examples have been asked. Look at the years. So all the examples have been asked one or the other point of time. So what is analogy? Analogy will have different origin, different structure, but common function. So how do you find out whether it is analogy or homology? So analogy is indicated by common function. For example, see, both wings, be it of butterfly or birds, it will help in flight. Clear? So this results in convergent evolution. Second, flippers of penguins and dolphin. Penguin is a bird, dolphin is a mammal, right? But both are involved in swimming. Then sweet potato and potato. Sweet potato is a root modification, while potato is a stem modification, but both help in food storage. Are we clear? Are we clear till here? Then we have some biochemical similarities also. Biochemical similarities also indicate evolution. Is that clear? Please let me know, guys. Another interesting observation supporting evolution by natural selection comes from England, and we are talking about industrial melanism, where two types of moths were studied: the dark colored moths, or the melanized one, and the white colored moths, right? So in before industrialization, it was observed that white were more than the dark one, while in 1990 the dark ones were more as compared to the light ones. Why? Because before industrialization there were lichens. Since there was no pollution, lichens were covering the bark of the tree. I hope that is clear, right? And in order to camouflage, these white ones were better. So white ones were not preyed upon. Rather, dark ones were easily visible to the predators. Is that clear? So that concept is explaining. So predators were able to spot a moth against the contrasting background. Is that clear? So lichens can be used as the industrial pollinators. We already know this. Is that clear? Now, second example of natural selection is excess use of herbicides and pesticides and the selection of resistant varieties. Now, this is due to anthropogenic action. This has been asked. Similarly, other cases of natural selection by anthropogenic action. One is this herbicide resistance or pesticide resistance. Second is drug resistance, and then man-made dog breeds. These are all through anthropogenic action. Now, talking about adaptive radiation, this was studied by Darwin in case of Darwin finches in Galapagos Islands. What did he observe? See, this represents adaptive radiation. Question was asked in 2023. Now, Darwin finches represent one of the best examples. Now, there is this question: what were the original finches feeding on? Original finches were seed-eating variety. Original finches were seed-eating variety, but later on they became insectivorous and vegetarian as per the availability of food. Another example is of Australian marsupials, and you have to learn this set of wait a second, guys. You have to learn this set of examples. Where is this chapter? Yeah. Please come here. So I hope you know that there is this Australian marsupial radiation given. Let's wait for it to come back. Till then, so other than adaptive radiation, we have adaptive convergence also. So adaptive convergence is observed here. Adaptive convergence here. Call it convergent evolution. See, this is adaptive convergence. So you have to learn them in pairs also. Also know that which one are placental and which one are Australian. So placental mammals are, see, mole, anteater, mouse, lemur, flying squirrel, and then we have bobcat and we have wolf. And in case of Australian marsupials, we have marsupial mole, we have numbat, and we have marsupial mouse, spotted cuscus, flying phalanger, and Tasmanian tiger cat. And you have to learn them in pairs also. So there are different types of questions that can come from here. Number one, you can be asked which of the following set belongs to the Australian marsupials. You can be asked which of the following set of animals belongs to placental mammals, and also which of the following sets exhibit convergent evolution. So there are three different types of questions that can come up. Is that clear? Now, biological evolution, the essence of Darwinian theory. What does Darwinian theory belong to or believe in? Evolution through natural selection. Is that clear? Then, so that is why, see, branching descent and natural selection are the two key concepts of Darwinian theory of evolution. So see, according to him, nature selects for fitness. So fitness, and this fitness is inherited. Is that clear? There must be a genetic basis for getting selected. So that is this. So adaptive ability is inherited basically, and it has genetic basis. Fitness is the end result of ability to adapt and get selected by nature. Is that clear to all? Please tell me these points are clear to everyone. Are these points clear to everyone? Yes, please. Are these points clear to everyone? How many of you are going to appear for NEET 2027? Is there anybody who's appearing in NEET 2027? Yes, please. Jamie. Jamie is for NEET 2027? And Akshay. Is that so? Just write on 2027. Maria. All right. Just write on 2027. All the 2027 students here. Yes, please. Amog. Very good. All right, guys. All right. Cool. Now, coming here. So, Darwin. Even before Darwin, there was Lamarck, a French naturalist, Darwin, who believed that evolution of life had occurred driven by Lamarck believed in use and disuse of organs. He believed in this use and disuse theory and explained the example of giraffe. How did giraffe acquired elongated neck? So this happened over generations. It was not overnight thing. It happened over generations. So to forage the leaves on tall trees, they had to adapt by elongation of their necks. So elongation of neck in giraffe, this thing is explained only by Lamarckism. Is that clear? This is explained only by Lamarckism. Nobody believes this conjecture anymore, but we don't have any other explanation for this as of now. Now see, what about Darwin? Darwin was influenced by Thomas Malthus. Darwin was influenced by Thomas Malthus. Yes, please. Quick. All right. Natural selection is based on certain observations which are factual. What does natural selection? It is based on that resources are limited, so populations are stable in size except for seasonal fluctuations. And in order to survive, they developed variations also. Is that clear? And these, so population sizes in reality are limited because of the competition for resources. Is that clear? So he asserted that variations which are inheritable make resources utilization better for the few and enable only those to reproduce and leave more progeny. Now talking about mechanism of evolution. Mechanism of evolution was explained by Hugo de Vries, and according to him, mutation leads to evolution and they are random and directionless, while de Vries believed that mutation caused speciation and hence leads to saltation, single step large mutation. So see, these questions are also come up in different years. Now talking about Hardy-Weinberg principle, allelic frequency in a population is stable generation after generation, but for this there are certain criteria. The gene pool or the total genes and their alleles in the population remains constant. Sum total of all the frequencies is 1. 1 means constant. So see, this graph represents different types of natural selection. When the peak gets higher and narrower only in the center, so that is when medium-sized individuals are favored. So peak gets higher and narrower towards one side, this side or this side. This indicates This indicates directional type of natural selection, while this is normalizing or stabilizing. While when there are two peaks and both the extremes are favored, this is called disruptive. This is called disruptive. Is that clear? So be sure that you are able to identify these, and on these on this concept we get some numericals also. You will be given 3 values and you have to find out on the basis of the percentage of the values whether they belong to disruptive type or the stabilizing type of the directional type. Now coming on to this, we have frequency. How is frequency of heterozygous individuals expressed? So first of all, this Hardy-Weinberg principle can be mathematically expressed as P plus Q is equal to 1, and if we square it, it becomes P square plus Q square plus 2PQ is equal to 1. Now what does P and Q indicate, guys? P and Q represents the frequency. So P and Q represents the frequency of allele, dominant allele and the recessive allele. While here P square represents the frequency of dominant individuals, homozygous dominant. So P square indicates the probability of dominant individuals. And then we have AQ, that is 2PQ. 2PQ represents frequency of heterozygous. Is that clear? Now what is this 5 factors which disturb the Hardy-Weinberg equilibrium? Number 1, gene migration or the gene flow. Second is genetic drift. Third is mutation. Fourth is recombination. And 5 is natural selection. So genetic drift operates in small isolated population. Yes, in small isolated population you can see the changes occurring very quickly. This is an important line. Think about it. And then new genes or alleles are added to the new population and these are lost from the older population. Gene flow. If this migration, there would be gene flow if migration happens multiple times and leads to genetic drift. What is genetic drift? It is change in the allelic frequency. Now the drifted population will become the founder population and leads to founder effect. Is that clear? Then directional changes, that is more individuals acquire value other than mean. This is known as the this is known as the directional natural selection. In stabilizing natural selection, more individuals acquire the mean value, that is the intermediate value. This is the value other than mean, it is directional. And if it acquires the peripheral characters, value at both ends, this is called disruptive. So this is how different types of natural selections are explained. Now brief account of evolution. So earliest organisms were non-green and anaerobes. First autotrophs were chemoautotrophs who never released oxygen. See here. So the first cellular forms of life about 2000 million years ago. See this one is also important. This one is also very important. This came in 2024. So you should know, when did mammals originate? Mammals originated into Cenozoic era, birds and reptiles in the Mesozoic era, fishes and amphibians in the Silurian one, and lower invertebrates in Proterozoic. Then about these plants, when was seed ferns originated? In the Carboniferous period. Ferns, conifers, cycads in the Jurassic period, right? Then ginkgos, nettles and dicots in the Cretaceous period. So this is how you have to learn. So first you should know the order of this era, right? Then in which era what event has taken place. Is that clear? Then see, South Africa happened in 1938. A fresh fish caught in South Africa happened to be a coelacanth, which was thought to be extinct, but these animals are called lobe fins. And what are lobe fins? Lobe fins are connecting links between fish and amphibians. So coelacanth is a lobe fin and it is a connecting link between fish and amphibians. Is that clear? Now coming here on this figure. So this figure is also important. So early reptiles gave rise to sauropsids and synapsids. And who came first? Synapsids came first and then came the sauropsids. Synapsids gave rise to Pelycosaurus, which are extinct, therapsids extinct, and then the mammals, while sauropsids gave rise to all these other reptiles and birds. Is that clear? Is that clear? So this is from synapsids, and then sauropsids gave rise to other reptiles and birds. So that is why we say reptiles gave rise to birds and mammals. So there was a parallel evolution taking place. So first amphibians that lived on both land and water was lobe fins. Is that clear? Now coming here to the origin of man. So this question has come up again and again, stages in human evolution, and second regarding their cranial capacity. So first of all, just know their stages. First was Dryopithecus, then Ramapithecus, then Australopithecus, then Homo habilis, erectus, and Neanderthalensis and Sapiens. And see, match the following for the cranial capacity. So the cranial capacity of Dryopithecus was 350 cc, Ramapithecus was 350, this was around 500, Homo habilis was 650, erectus was 900, Neanderthal was 1400, and Homo sapiens was 1350 cc. But in between Neanderthal and Homo sapiens actually came the most intelligent, that was Cro-Magnon, who had the highest brain capacity, which was around 1650 cc. This was the most intelligent of all. Homo sapiens fossilis, the immediate ancestors of humans. Is that clear? So guys, this was evolution. So make sure that you learn these points. I will be uploading all the PDFs today in the Telegram channel. Make sure that you follow me on Telegram channel, and my Telegram channel is, wait, I'll write down here. Tina Ma'am Biology. Is that clear? All right. Let's move on to the next chapter, guys, that is health and diseases. So earlier we used to believe that it is a balance of certain humors which kept the body healthy. This is according to Ayurveda, the humor hypothesis. But Hippocrates and Indian Ayurveda both believed in this humor hypothesis. This was known as humor hypothesis. See, Hippocrates is the same person in the name of who you take an oath after MBBS. So it's oath of Hippocrates. But later on, William Harvey discarded it. So Hippocrates and Ayurveda believed that a person with a black bile belonged to the hot personality and causes fever, right? But later on, William Harvey, he just discarded this theory at all. He disapproved it, disapproved this good humor hypothesis, right? And according to WHO now, according to WHO, what do we believe? That health is a state of complete well-being. So WHO has brought together so many good things of so many different scientists, right? So mind and mental state can both affect the health. What are the different factors? Genetic disorders, infections, and lifestyle. Now, is that clear? Coming on to, so we have two different types of diseases here. One is infectious, other is non-infectious. So we know that infectious diseases are because of some or the other pathogen, right? So pathogens are the causative agents. It can be bacteria, fungi, protozoans, helminths, and virus. Coming here, Salmonella typhi caused by bacteria, is a bacteria, causes a disease known as typhoid. And what happens in typhoid? So it affects which part? Small intestine. How does it enter? Through contaminated food and water. What happens? Sustained high fever, 39 to 40 degrees Celsius, weakness, stomach pain, constipation, headache, and loss of appetite. Is that clear? So extreme cases, in severe cases, intestinal perforation and even death. It is confirmed by what is a diagnostic test? It is Vidal test. Typhoid Mary, classic case of typhoid carrier. She was a cook by profession. Another bacterial disease is pneumonia. We have already read Streptococcus pneumoniae, also caused by Haemophilus influenza. The question has been asked multiple times. Now, it infects your alveoli basically. It affects your alveoli. So alveoli filled with fluid, leading to several problems, include fever, chills, cough, and headache. Then body turns, lips and nails turn gray to bluish in color. And it occurs due to droplet or aerosol infection and also by sharing of utensils and glasses. Then other bacterial diseases are dysentery, plague, and diphtheria. So what are the bacterial diseases? You have to learn the name of the bacterial diseases based on the pre-vi cues. Now see, viral disease, rhinovirus. Rhino word is related to nasal epithelium. It causes common cold and infects nose and the respiratory passage, but not the lungs. Remember that. How is it symptomized? The symptoms are congestion, discharge, sore throat, hoarseness, cough and headache and tiredness. Lasts for only 3 to 7 days and then gets treated itself. Then how is it picked up? Like, so that means what is the mode of infection? The mode of infection, it is inhaled directly, that is droplet or through contaminated objects. Then let's talk about the protozoan disease, that is malaria, number 1, caused by Plasmodium. The different types of Plasmodium are Plasmodium vivax, malaria, and falciparum. This is the most dangerous as it causes malignancy and the most serious one and will be fatal. Is that clear? Now, how does Plasmodium enter the human body? It enters the human body in which form? Sporozoites. Hence, it is the infectious form. Through the bite of female Anopheles mosquito. It multiplies within the liver cells and then attack the RBCs, resulting their rupture. Now, rupture of RBC occurs due to hemozoin. Is that clear? So, when hemozoin is released, it causes rupture and that's when the malarial attack sets in. Is that clear to everyone? Now this parasite requires 2 hosts, hence it is digenetic. One is the female Anopheles, which is a vector, and the primary host, and human is the secondary host. Why primary host is female? Because it shows sexual stages or the sexual cycle, and human has the asexual stages. So the one with sexual stages is known as the primary host. Is that clear? So this is the cycle. So it starts from here. Mosquito bites another human, sporozoites injected with the bite. Sporozoites reach the liver through the blood, and this is where parasite reproduces asexually in the liver cells, bursting cells and releasing them into the blood. From the blood, guys, the parasites reproduce asexually. In second reproduction occurs in RBCs. When burst the RBCs, then the cycle of fever sets in. Is that clear? And it also infects the new RBCs. That is why it leads to anemia. Then sexual stages are developed in the RBCs. Very important. Gametocytes develop in RBCs. So all these phases are occurring in the secondary host, that is human. Now, when a mosquito sucks blood, it is going to pick up these stages. Which stages are picked up? Gametocytes. Gametocytes. See, female mosquito takes up gametocytes with the blood meal, which will undergo meiosis. So meiosis will occur and will form gametes, and they will undergo fertilization. Where? In the gut region. So in human, it is partially present in liver and otherwise present in RBCs. While in mosquito, which is the primary host, it occurs in the gut region, gut area. Is that clear? And later on in the salivary glands, these sporozoites are stored. So mature infective stages, that is sporozoites, escape the gut and migrate into the salivary glands. So this is the cycle of malaria. Another protozoan disease is Entamoeba histolytica. So every year we are getting one or the other disease along with the causative agent and the symptoms. See, what are the protozoan diseases that you should know? Is that clear to all? So it is leishmaniasis, trypanosomiasis, amoebiasis, and even malaria. Now, in Entamoeba histolytica, amoebiasis is going to cause amoebiasis or amoebic dysentery, constipation, abdominal pain, cramps, stool with excess mucus and blood clots. Housefly is the mechanical carrier. Who's the vector? Housefly. Right? Then, talking about, so drinking water and contaminated food by fecal matter is the main source for amoebic dysentery. Ascariasis, the common roundworm. Now, this is a helminthic disease that we are studying. It leads to it leads to ascariasis, two diseases which are occurring by helminths. One is ascariasis, another is filariasis. These are the two diseases we have to study, helminthic or ascalminthes. Now see here, wuchereria. First, let's talk about ascariasis. Ascariasis include internal bleeding, muscular pain, fever, anemia, and blockage of intestinal passage. Again, contamination through soil, water, and plants, and occurs through contamination of the food. Then wuchereria, bancrofti, and malaria leads to chronic inflammation of lymphatic vessels of the lower limbs, leads to the disease elephantiasis or filariasis. Is that clear? The question has been asked multiple times. So it causes blockage and hence lots of swelling. Then fungal disease, the three genera Microsporum, Trichophyton, and Epidermophyton. So question around Microsporum and Epidermophyton has already been asked multiple times. Maybe this is the turn for Epidermophyton, right? It is known for ringworm infection, the most common infectious disease in humans. So dry scaly lesions on skin, nails, and scalp, right? Heat and moisture is the best condition for them to thrive. Is that clear to everyone? So these were the infectious diseases. Now coming on to immunity. So first of all, we have, see, immunity is of two types. Innate is one that you're born with, and acquired is something that you acquire during the lifetime. So let's see here, innate, it is non-specific, present since time of birth. Question has already been asked. So what are the four barriers here? Physical barriers. It includes skin, it includes mucus, right? And then we have cilia also here, which are going to push this infected mucus outside. Now, physiological barriers includes acid in stomach, saliva in mouth, tears in eyes, then wax in ears, and lactic acid in vagina. Cellular barriers, guys. Cellular barrier contains some phagocytic. They contain some phagocytic cells. What are the phagocytic cells? WBCs. Not all, but neutrophils, also known as polymorpholeukocytes, that is PMNL, then monocytes and NK cells. Is that clear? They all constitute cellular barriers, but phagocytes are only PMNL and monocytes. These are not phagocytic. And the third phagocytic cells are macrophages, 1, 2, and 3. But when we talk about cellular barriers, then natural killer cells will also be a part of it. Natural killer cells will take care of the tumorous growth in your body. Then cytokine barriers include interferons. So virus infected cell is going to secrete these interferons to protect the non-infected cells from further infection. This is also an example of innate immunity. These questions were asked in the form of match the following. Is that clear? Then let's talk about acquired immunity. See, this was all in 2022. What is the difference? Acquired immunity is always pathogen specific. It develops when body encounters a pathogen for the first time, and at that time the immunity is known as primary immune response, while subsequent encounters will generate the secondary immune response, also known as anamnestic immune response. Is that clear? Now it is ascribed to the fact that body appears to have memory for the first encounter. So memory is one very important characteristic of acquired immunity. So acquired immunity is characterized by specificity, written specificity. Second is memory. Third is diversity. So as many antigens are going to enter, our body can produce that many antibodies. And last not the least, it can distinguish between self and non-self. Self and non-self. It can distinguish between this. Is that clear to all? Now, there are two types of lymphocytes, B lymphocytes, T lymphocytes. B lymphocytes are meant for producing army of proteins, that is known as antibodies. Army of proteins against a pathogen. These proteins are called antibodies. T cells don't produce antibodies, and each antibody has 4 polypeptide chains, 2 light chains and 2 heavy chains. This was asked in 24, this was asked in 25. This whole structure based question was asked in 25. See, this side on the N terminal we have antigen binding site, and towards the C terminal we have constant regions. 4 chains are held by disulfide bonds, right, between the light and the heavy chains. Is that clear to all? Now Talking about CMIS, there is another type of lymphocyte, T lymphocyte, which produces cell-mediated immune system, CMIS. So some human organs like heart, eye, liver, kidney fail to function satisfactorily. So when transplantation occurs, right? So it is CMIS, cell-mediated immune system, which will ensure whether that transplant will be successful or not. So why do we need to check for the compatibility? Because otherwise cell-mediated immune system is going to reject it. See, so cell-mediated is responsible for graft rejection. The body is able to differentiate between self and non-self. So I told you already, this is a characteristic of your immune system. Is that clear? Now, similarly, tissue matching, blood group matching are essential before undertaking the graft, after which the patient has to be taken for. Otherwise, also, patient will be taking some immunosuppressants. Please tell me all these points are clear to you. Yes, guys. Please tell me all these points are clear to you. Yes, please. All right. Now, moving on to active and passive immunity. See, when a host is exposed to antibodies are produced into the body. This type of immunity is called active immunity. When the body is able to produce antibodies on its own, that is active. Active immunity is slow but takes time to give its full effective response. Now, when ready-made antibodies are directly given against the foreign antigen, it is called passive immunity. Yellowish fluid, colostrum, secreted by mother during the initial few days of lactation are abundant, has abundant antibodies. And which one? IgA. Fetus receives some antibodies directly through mother's placenta during pregnancy. These are both examples of passive immunity. Please tell me these points are clear. Now, talking about vaccination and immunization, see, it is based on the principle of memory. It is based on the principle of memory. What is vaccine? Vaccine is inactivated or antigenic proteins of the pathogens which are introduced into the body, right? Now, antibodies are going to neutralize these, and also vaccines generate memory. See, that is the principle of this. Is that clear? Now, in case of snake bite, injection which is given to the patient contains pre-formed antibodies against the snake venom. That is why any vaccine is an example of active immunity. Remember that, because you are just injecting the pathogen, antibody will be produced by the body itself. Clear to all? Now, but when we are directly injecting the antibodies, this is called passive immunization. This is known as passive immunization. So guys, there is some short circuit. Wait a second. There was a fire near our place. 5-6 shops have been damaged, and there is some issue outside. Yes, guys. I think there is a storm outside, because of which there is an issue. Let me check. Okay, yes. Yes, there is a short circuit because of the storm outside. So I think we'll have to wind up here. I have already completed ecology during my marathons. I've read the NCERT. As far as these two chapters are concerned, biotech and this one, I'll be putting some short videos regarding the chapters, and make sure that you watch that so that we are able to complete the NCERT reading. Is that clear? So we'll have to stop this session here. But don't forget to solve these questions with me in special classes tonight. Okay, in all the special classes, and I'm going to take that tonight also at 9 p.m. I'll see you later. Thank you so much for joining me, and don't forget to watch these special videos now. And also in the coming session, I am going to discuss some most important points, guys, which is required for your last minute revision points. Because see, remember, you do know lots of things. There's so much in your head. Have that belief that you will be able to recall these things at the right time. They will come back to you. But then there are certain things which are factual and data based, which we need to revise till the last moment. So I'm going to do that with you in the coming session. By the way, tomorrow at the same time we are going to meet for revising all the diagrams. Is that clear? So I'll be revising rest of the things in that session also. So make sure that you watch that session tomorrow. We'll be discussing and revising all the diagrams tomorrow at 12. Bye.